So hi, my name is Megan and I am a scientist. So most days you'll find me here. This is me in my lab wearing a white lab coat and surrounded by bottles of chemicals and jars of pickled animals, doing amazing experiments like this one. I'm levitating this tower of eggs using my brain power alone while my adoring student watches on. Okay, that's a stereotype. This is my lab and I do have a white lab coat and I do have adoring students, I hope. But this doesn't really describe what I do as a scientist. These are the tools that I use, but what do I really do? So being a scientist is amazing because it gives me license to roam and explore the boundaries of human knowledge. So I like to think of human knowledge like this, as a golden globe floating in a sea of dark unknown. And if you look to see where I am, then I'm here. I'm right at the edge. I'm right at that interface between the known and the unknown. So that's an extremely dynamic place to be. That's my coalface. That's my workplace. That's where I think of ideas and test hypotheses. That's where I take small chunks of the unknown. I take them close, I study them, I try and simplify them, I try and understand them, and at some point I hope that I add them to that pool of human knowledge and push that boundary just a little bit further out into the unknown. And what an amazing time to be a scientist and working in that place. Right now, I truly believe that we are in a golden age of science. There are a huge number, more than ever before, people working on scientific problems around the world. So as an example of this, since 2000, we now spend 3 times more around the world in research and development. And in terms of understanding and knowledge generation, we also publish about 3 times more papers per year now than we did in 2000. So this year, we'll probably publish 1.8 million scientific publications. But the reason that I think this is truly a golden scientific age is that for the first time in history, this knowledge doesn't just belong to us academics or to us scientists. This is actually for the first time your knowledge, and it's yours for the taking. So now the movement and the reason that this is really your knowledge is because for the first time it's available to you. Free and open access and free of charge. So this is a movement that's been driven by many different organisations, for example, in the UK by Research Councils UK and by the Wellcome Trust. And they stipulate that any research done that they've funded must be freely available to the public. And so it's there online. And Research Councils UK estimate about 75% of papers supported by them are now freely available. So, you know, this is just an amazing time, and my talk today is really about how you can access this knowledge and how some of this knowledge is there for your taking, and some of the research that I'm going to talk about today and how it's changed people's lives. But really, this is your knowledge. You've paid for it as well. So your taxes, they pay for my research grants. If you went walking down the prom as part of the moonwalk and blistered your feet, you were supporting someone's research grant somewhere. And if you took all your second-hand books and clothes down to the British Heart Foundation charity shop on the high street, then you were the one who supported my PhD, and so thank you very much. Okay, so what do I actually work on? I work on these. I work on your hands. So hands are amazing things, so just have a look at your hands for a moment and contemplate them, because everybody forgets about them unless they're really cold or really dirty or they've just slammed them in a door by mistake. So hands are fundamentally human. You know, we use our hands in a way unique to all animals. We use them to manipulate our environment. We use them to communicate with each other. We use them to show love and caring. We also use them to fight. So people can look, believe that they can look into your hand and that they can see your future. Well, okay, I'm not that clever, but I can look into your hand and I can see something of your embryonic past. So the 2 questions that I'm really interested in working on are, why do we have 5 fingers? And also, how do these fingers all have a different identity? So on one side you have a thumb, and on the other side you have a little finger. So the type of scientific experiments I do are really, in a way, not so different from what these prehistoric people were doing. I look at how patterns form in the developing embryonic hand, and to do that I use model organisms, particularly from birds, so ducks and chickens and emus. So this is the type of pattern that I like to look at. So this is a developing chicken foot. And the dark blue there is a gene expression of a gene called SOX9, which will tell those cells in which it's expressed to form cartilage. And as the dark blue signal goes off, then the light blue signal, which is the cartilage, will begin to develop. Right, so back to this question of 5 fingers and different identities. Now there's really one gene which controls this, and if you remember nothing else about what I've said today, you'll probably remember this. So this is a chicken embryo, and it's showing expression of a gene called sonic hedgehog, and that's critical about what I'm researching. So everything that's dark blue here is where sonic hedgehog is turned on. And if you can see on the side of the body, there's 2 little lumps. Those are the developing wing and foot. And you can see on one side there's a dark area of cells, and those are the cells that are turning on sonic hedgehog. So the idea is that the cells which are very close to those blue cells and the cells which are actually making the blue signal, sonic hedgehog, they hear that sonic hedgehog signal very loud, and they're told to turn into little fingers. The ones on the other side of the limb, they're far away from the signal, they can't hear it so well, and so they turn into thumbs. So if you don't have any sonic hedgehog, you just develop thumbs. If you turn on sonic hedgehog for too long or in the wrong place, you end up with a lot of extra fingers and maybe no thumbs at all. So having 5 fingers is really down to sonic hedgehog being turned on in those precise cells for a precise amount of time at a precise level. And if you turn on sonic in the wrong time, then bad things can happen. So sonic hedgehog, though, is important in hands, and the reason I use hands is because it's a nice, simple thing. It's just five outputs, really, to look at. But sonic hedgehog is important in almost every organ that's developing. So every blue cell here is, at some point in its life, turned on a little bit of sonic hedgehog. It's important in almost every organ that's developing. And it's turned on right now in you. You've got sonic hedgehog turned on in your gut as it's regenerating, and probably in your liver if you're recovering from your stag do over the weekend. So when sonic hedgehog comes on in the wrong time and the wrong place in adults, then it can cause cancer. It's one of the main causes of skin cancer and bladder cancer and pancreatic cancer. So this is an example of when sonic goes bad. This is the TEDxPortobello chicken mutant. The one on the right is a normal chicken embryo, and the one on the left is its sibling. And I was lucky enough to come, when I first came to Edinburgh, to work at the Roslin Institute. I worked in a lab with Davey, and his team were looking to see what the genetic cause was of this chicken mutant. Now, we were very lucky in that we were able to find what the gene was that was abnormal, and we called this the TEDxPortobello gene. It was a gene that no one had ever worked on before. Now, when we found this, it was just after the first human genome had been sequenced, which meant basically we had a list of all of the genes that are in humans, and we could compare straight away and to show that there was a TEDxPortobello gene in the chicken that we could show was important for embryonic development, and there was a TEDxPortobello gene in humans as well. But we didn't know if that would be important for human development. So I took 10 years, and I've been looking at what the TEDxPortobello gene does and how the cells use it to hear how much sonic hedgehog they should be seeing. And in that 10 years, as well as understanding what was happening, how TEDxPortobello works, there's also been a revolution in genomic technology and in biology. And that's down to sequencing genomes and the speed at the cost at which genomes can be sequenced. So that first human genome that was sequenced, that cost $3 billion US. Today, it costs £600 to have your genome sequenced. So this means that genomic sequencing is now used as a diagnostic tool around the world. Now, we predicted from this chicken, which is embryonic lethal, it doesn't survive, that probably the TEDxPortobello gene would be so important in humans that humans with 2 bad copies would probably not survive either. But in the last 10 years, there have been hospitals and doctors around the world which have seen families, very few and very rare patients who have 2 bad copies of TEDxPortobello, and it seems that sometimes one of these copies has a tiny little bit of function, and that allows that patient to have life. So what happened in this kind of genomic revolution is that in isolation, none of those clinicians would really be able to diagnose their patients as having a TEDxPortobello mutation in isolation. But because we have all of this genomic data available to everybody, those doctors and clinicians were able to compare notes. They were able to read my research papers, which were online for free, and realise that this was an important gene. And so slowly the pieces of the puzzle have come together, and we've realised that TEDxPortobello is a really important gene in human development too. Now, what's amazing is that not just scientists and doctors have been accessing this information, which is out there for free, but also just ordinary people like you and me. So this is a family which contacted me last year. They had a daughter who had been born premature, and she had some unusual kind of developmental problems. In particular, she had poor motor control and she couldn't breathe very well, and her eyes would flicker quite a lot. Now, they'd had their genome sequenced, which I already think is kind of amazing, but the report had come back as negative. So the idea, there was no obvious genes that the doctors could see in this report that had gone wrong. But that was because at the time, there was no published literature to show that the TALPID3 gene was actually important in humans. We were still preparing that paper. But the mum didn't stop there. She actually took that genetic report, she read it herself, she realised that her daughter had 2 bad copies of TALPID3, and then she went into that pool of literature and she investigated all for herself. She read all of the open access papers, she read all of the stuff about the chicken, she read around the subject, and she realised that there was a spectrum of disorders, and perhaps her daughter had these problems because she had mutations in TALPID3. And so eventually she contacted me and asked what I thought. Well, luckily I already knew of a few other patients and they had exactly the same symptoms as her daughter. Now, these patients all have a syndrome called Dubier's syndrome, which is characterised by a very small malformation in their brain. And so over an email conversation, only over a few hours, I suggested that she should go back to her doctor and ask for this MRI scan. The mum actually already had the scan. She had it on a computer, and a few minutes later she sent me back the correct section, the correct image of her daughter's brain, and together we realised that her daughter probably did have Dubier's syndrome. So the great thing was she went back to the doctors and they confirmed the diagnosis. The family is now supported by Dubier's syndrome support groups, and she's in contact with clinicians in America who specifically work on this syndrome. And you can see what happened next. They had another baby, and luckily they were able to do a genetic test on their second child to see whether he would have Dubier's syndrome or not before he was born. And thankfully he didn't, and he's a very happy and healthy little boy. So, you know, that's an amazing story. Full credit to her. She took that globe, that orb of human knowledge, and she investigated. She was living in this, you know, with a situation where she was just crowded with unknowns. She didn't know if her daughter, why her daughter was ill or what the proper treatment would be. And so she went to all of that literature, all online for free, and she found out for herself. And that's been really fundamental to how her daughter's treatment goes forward. So just back to this human knowledge. Now, the boundary which that's expanding out into the known is unstoppable. It's expanding massively and quickly every day. And people get really scared, actually, about how this new technology is going to be used and you know, what our scientists are doing. Well, I'd just like to say that as we learn more, we can see a little bit further out into the unknown. And I can tell you that out there are cures for diseases that we know about. In fact, there's probably cures for diseases we don't even know about yet. The point is that scientists, we can't really determine how we use our new technologies. It's up to society and for scientists and society to have a dialogue together. So I really say, don't be scared of what's out there in the unknown. It's extremely exciting. Replace the fear that you might have of how this technology is going to be used instead with a curiosity. And I say, as a scientist, to take my hand, let's go together into the future. All of this information is at your fingertips. It's there for the taking. So grab it and use it. Thank you.